All right. Hopefully, we will be studying for a while this time. Um, this is also a very quite risky video. I feel like because we're gonna be going through like 3 textbooks in this one video, and I don't know if that's allowed on YouTube, but I don't really care. Um, so before we begin, this is obviously a video on plants, but the thing is, I would say since it's confusing. Okay, there's a lot of different things. If you just look at this cross section of a leaf, we're seeing first of all this is a leaf, and a stem is a separate whole matter. We're looking at vascular systems. Okay, you're looking at the xylem, you're looking at the phloem, you're looking at the parenchyma and the specialized versions of that. Okay, but then when you look at the leaf, you have a lot of things to work with. You have your don't start with the organelle first. First, you have different types of cells. You have your palisade mesophyll, spongy mesophyll. Sorry, upper epidermis, palisade mesophyll, spongy mesophyll. Then you have, of course, going to be your vascular bundle going through somewhere around here. You're gonna have, as you go down further, you're gonna have stomata between your epidermis cells. Epidermis is going to specialize into stomata. Then you're gonna go into even more specialization. We're gonna see your midrib of your leaf, right? You're gonna have your leaf, and you're gonna have a cross section of that. You're gonna see, you know, since this middle part is gonna be the most, it needs to be the most structurally stable to hold the entire leaf. So you're gonna see the structure. You're gonna see the where there's gonna be more sclerenchyma and stuff like that. So there is a lot of stuff, okay, and other textbooks will emphasize more. Such as this textbook starts off with the cell wall. Why would you start off with a cell wall? God knows, right? Because cell wall is not even it's not even a cell. It's an organelle. We're starting off with the organelle. So it is not very clear, at least to me, um, because they are really working off of 3A2 and 3A3 in that Excel textbook. So, I mean, everything aside, if you just look at your specification for biology, what do they truly want? Forget about the textbook and extra information there. What do they truly want? They truly want, regarding that kind of stuff. Mm, so I remember going through this a long time ago. I don't know why I wrote Solanum over there. I don't know why else people know. A lot of drawings here, I'm sorry. So it's just structure and ultrastructure and then function of the stuff, and we're talking about the organelles. Okay, so we're talking about cell wall, talking about blah blah blah. So we're talking about basically ultrastructure first and structure, as well as in structure of the leaf and then ultrastructure of the cell, then functions of all those things. So this is kind of starting off as almost the same thing as gonna be your ultrastructure of animal cells. So we're kind of doing the same thing here. Then we're doing organelle, just being able to recognize, same thing as last time. Structure function of polysaccharides. So now we're looking at what's going to be present inside the cell walls. So it's going to be cellulose. Mainly just talking about relationship of how do the beta glucose works, and also this is important, the cellulose microfibrils, how those are involved. Yeah, exactly, how cellulose microfibrils and the secondary thickening in the cell walls, these two things, physical properties of xylem vessels. Okay, now we're going to be looking at sclerenchyma, xylem vessels, and phloem. These three, and then we can talk about the rest later, because that's going to be going into smaller things such as plant fibers. I think is truly important. This is interesting. This I need to look into, and then this is just the smaller stuff like how they survive, conditions for bacterial growth, antimicrobes, and stuff like that. So we're not going to go into that too quickly. So yeah, um, so that's kind of how they start off in this textbook. In this textbook, however, they go into that. I think they go into that a bit later. Um, first they, oops, okay. So first they talk about transport needs, but that's not really something we should be looking into. First, they actually talk about the stem or the vascular bundle here. They talk about the two systems. Then they talk about structures of each one at a time. So first they talk about the stem. Well, no, this is not the stem. This is gonna be. No, wait, that is your stem. No? It's gonna be your root. But anyways, it's all included here. Then they go into ultrastructure transport, and then they talk about specifically, stuff like this. Quite later on in the actual thing. Here are some examples of plans, adaptation wise. So that's kind of the thing. That's kind of where I'm confused, because it's almost as if they assume some certain knowledge, which I kind of find hard to visualize. Even this, like, this is just a bunch of text. So this is going to be our third textbook where they really visualize it. I really enjoy this. However, before we even begin with all of that, I did want to go into. Oh wait, you can't see this textbook, can you? It's okay, I can make it work. So that's how it works. Very interesting. So now you can see my desktop. Okay. Anyways, so this is kind of what I wanted to look into. It's a bit far off because now we're looking at pretty much animal stuff. But cytokinesis, mainly I wanted to delve into the plant cytokinesis, because then we can learn actually about the cell plate, which will tell us a little bit more about the cell wall. Okay, so there's gonna be a lot of words I don't know here, and I'm just kind of ready to just skip those because this is a uni textbook. So yeah. So in animal cells, cytokinesis will happen through a process called cleavage. Okay, and I do remember this word in our own textbooks. The first sign of cleavage is the appearance of a cleavage furrow, which is a shallow groove in the cell surface near the old metaphase plate. So they have it all in this one diagram. 12.10, it's actually in this diagram. There you go. So in the area of the old metaphase plate, right? Metaphase being obviously the thing where they line up in a straight line. So in that same exact area, you're gonna see a cleavage furrow, which is it going in words, of course, down like a contour. You can see each contour. This is almost as if you can see it's got clothing. Okay, so you're gonna see the appearance of that cleavage furrow, and okay, this will lead into plant cells. I feel, but anyways, I'm learning. So shallow groove in the cell surface near the old metaphase plate. So where we have that old metaphase, we're gonna start seeing a cleavage furrow forming. Now, on the cytoplasmic side of the furrow is the contractile ring of actin microfilaments associated with the molecules of the protein myosin. So, what's happening here? On the cytoplasmic side of the furrow. So, what does it mean by cytoplasmic side? No idea. But there are filaments attached to it due to the myosin protein. The filaments react with the myosin molecules, causing the rings contract. So, okay, I do remember this. So when you have this and you have the furrows, you have filaments, microfilaments, and when they react with myosin, then they'll start to contract these, pulling these apart. So it's truly pulling the cell apart, really. It's not really pulling the things apart. Or perhaps it is. Oh, God. I've forgotten my mitosis. Dang. Do we have a mitosis picture? A mitosis picture would help. There you go. So you have your metaphase plate, you have your anaphase, and you have your telophase and your cytokinesis. So anaphase is where these get pulled apart separately. Telophase is where the cell itself gets pulled apart. And as you can see, if you have a cell that's first like this, or rather like this, then and it's going to be divided along this area, which is, it's the same area as where the metaphase plate was, right? So it begins as a line curving around the contour of the cell as a cleavage furrow, okay? When they say cytoplasmic side, I'm not sure. Anyways, though, so the contraction of the dividing cell's ring of microfilaments is like the pulling of a drawstring. The cleavage furrow deepens until the parent cell is pinched in two. Pinched as in, it's like you have a force that's pulling it this way, you have a force that's pulling it this way. So this gets down, so it's pinching together until that pinch just pops out and you have two different cells. Or rather, this is a bit too wide. So you have just this one and just this one, and there's a gap in between, right? So it's pinched into, that's kind of the wording you want to be used, making two separate cells, each with its all sorts of stuff. However, in cytokinesis in plants, cell walls are present, which makes it different, okay? This is gonna be what makes stuff different. There is no cleavage furrow, and instead, during telophase, okay, what is telophase? Let's just quickly check. Telophase is going to be where everything is going to start reappearing, I believe. So after these two are divided, we have these just chilling on each side. The nucleus reappears, chromosomes become less condensed, remaining spindle microtubules are depolymerized, as in the things that pulled them back, they get depolymerized. And mitosis, the division itself is completed. The division of the nuclei into two genetically identical nuclei is finished. So mitosis was truly just, it started from here and ended from here, because now it's the division of the cell. It's not a division of the nuclei anymore. So in this case, when telophase will happen, the vesicles derived from the Golgi apparatus, because the Golgi apparatus will be present within the cell. Now a question can arise, what is the Golgi apparatus? Because people can forget that. If we go into our second resource here, which you should be able to see, let's just read on it a bit. So Golgi apparatus. I do remember reading through it a little bit. So, your Golgi apparatus. Mm-mm-mm. Yeah, I'm liking this. So, the Golgi apparatus will be fluid-filled flattened sacs, curved sacs with vesicles um, surrounding the edges. So, this is going to be over here, Golgi apparatus. It has vesicles surrounding it, and the Golgi apparatus will process and package proteins and lipids. Okay, so you're gonna have proteins, you're gonna have lipids, they're gonna be packaged up, they're gonna be processed. So whatever's inside is gonna be processed a certain way, and it's just gonna be there. Okay, and that's what it does. The did I do that? Where is that line from? Now, if I'm not wrong, these proteins and lipids which get packaged up get sent out in vesicles. Right. So you have the Golgi apparatus actually being used here. So the polypeptide, which is obviously going to be a protein, sent to the Golgi apparatus, then it's processed, right? So it's processed. By processing, we mean that it gets folded into 3D shape. So if it was, well, polypeptide, it means it's going just like this. It's your chain of polypeptides, perhaps some secondary structure, but there's no tertiary structure, right? Because this is just a chain. So, that chain is then it starts to be twisted and turned in 3D shape to form a 3D protein. So that could be a third tertiary structure, or perhaps even quaternary structure. The point being is, now that it's in this 3D shape, it gets packaged in a vesicle, specifically a Golgi vesicle, but it's a vesicle, which fuses with the cell surface membrane to release the enzymes on the outside. Now, of course, it needs to be 3D shaped to form the enzyme itself, right? Because enzymes are proteins, and it's in the vesicle, because the vesicle is actually used to ensure that whatever the vesicle is used to ensure that the protein in the cytoplasm area, it's enclosed in a vesicle, because it is truly an enzyme at this point. So it doesn't react, or it doesn't catalyze reactions within the cell, only after it fuses with the cell membrane over here and it leaves. Then the vesicle is destroyed because it fuses with the cell membrane, and the enzyme itself is released to the outside. It's extracellular now, and it can catalyze reactions on the outside solution, right? So if the vesicle in this case is used to prevent catalyzation or any reactions for what's inside the vesicle with what's outside. Now you might wonder, why vesicle? Why not anything else? Because vesicles fuse the surface membrane. We want the enzyme to be reacting with outside the cell, not inside. So what's one thing which contains stuff inside but not outside? That's going to be your vesicle, truly, because it fuses with the surface membrane. Anyways, that's going to be your Golgi apparatus use. So now when we go back into Campbell, right, and we're talking about cytokinesis in plant cells. Okay, sorry, this is just bothering me. Why is this opening up? Because this. Whoa. What is that? Okay. Interesting. Okay, I understand the issue now. Funny. Okay. There you go. Hopefully that wasn't. Hope that wasn't too bad. Okay. So, yeah. Now in telophase, right? Rather than, you know, rather than the, after telophase is over, rather than a cleavage furrow, um, being formed to divide these two into two separate ones, you're gonna have vesicles from the Golgi apparatus, of course, moving along the microtubules. So there are microtubules present from the sides, from I'm guessing the centrioles, to the inside. There's no centrioles in plant cells, if I'm not wrong. It's gonna be it's something PCAT or something like that. They're going to move the enzyme, the vesicles will move along the microtubules to the middle, right, to the plate, kind of, where they coalesce, producing a cell plate. Now remember, vesicles fuse with the cell membrane, so they have some property which allows them to kind of form a cell membrane, right? So when you have the two Golgi apparatus from two either sides moving in across the microtubules into the center of the cell, right? Then they're gonna form like a cell plate of sorts. Okay. Now the cell wall materials carried in the vesicles. So the vesicles carry stuff. What are carried? The cell wall materials are carried, and they collect in the cell plate as it grows. Right. So now as the cell plate, as you can see over here, right? It forms the cell plate, and as more and more and more cell wall materials are brought into the thing. Of course, these are two nuclei, because that's what's going to happen at the end of telophase. So you're going to have across these microtubules more and more cell wall materials will be added, forming a better cell plate, forming finally a cell wall between the two daughter cells. Now the cell plate enlarges until its surrounding membrane fuses with the plasma membrane across the perimeter of the cell, right? So again, you're just having this, you're having cell plate that's forming in between. Eventually the cell plate becomes very strong. Eventually the cell plate starts actually fusing. This is actually where your vesicles will actually start interacting with the cell membrane in these two areas. When we say the word fuse, we always think of vesicles interacting with the membrane. Now, the cell plate is the vesicles. Don't mistake that. They are one and the same thing. And now we use the term fusing because this is truly the only two places where these two things interact: the cell plate and the cell membrane. So now when the cell plate enlarges enough to actually interact with the cell membrane, it fuses along the perimeter. Now, as a result, you have two daughter cells, and each has its own plasma membrane. As in, you have your plasma membrane here, you have your plasma membrane here. And meanwhile, a new cell wall arising from the contents of the cell plate forms between the daughter cells. So, what does this mean? This part specifically. Right? Because what does it mean that a cell wall will be made along the perimeter of the cell? It sounds weird. Is it as easy as I think it is? Right. So over here, we're looking at your prophase. You're gonna see chromosomes condensing, nucleolus beginning to disappear, mitotic spindle fiber is forming. Discrete chromosomes are not visible. Aligned sister chromatids. Later in prometaphase, the nuclear envelope will fragment. Spindle is complete. The chromosomes attached to microtubules at their kinetochores are all at metaphase plate. So you're gonna see basically your chromosomes, condensed versions, attached to microtubules at their kinetochores, which is going to be on each side, and they're all lined up in the thing. Anaphase is when these are going to be separated. And daughter chromosomes moving to the ends, as well as their kinetochores microtubule shortened. Telophase is where specifically we are focusing on, is when daughter cell nuclei are forming on each side. Cytokinesis will start, in which the cell plate will form, which divides the cytoplasm into two. As in, imagine this is one cell originally, because it is one cell originally, right? And so the cell plate will start to form until it fuses with the cell membrane because it grows large enough, dividing the cytoplasm, this cytoplasm, dividing it into two parts, and it grows towards the perimeter. It grows towards the perimeter of the plant cell. I don't know what the sentence means. Does it mean it's like after growing this, it goes over here and it goes over here and this goes over here and this goes over here? Oh. Okay, so now that we're caught up a little bit on how that cell wall division actually forms, let's talk about what that cell wall actually is, right? Or rather, how that cell membrane forms between the two cells. Let's actually talk about what that cell wall actually is. Here are some stuff. So the cell wall itself is going to be the rigid structure right outside, adjacent almost to the cell membrane. Okay, it's a rigid structure that surrounds the plant cell, and it's mainly made out of cellulose. Okay, I feel like that's pretty much the important part, and it supports the cell. Okay, now here is something that's kind of confusing. So the middle lamella is going to be the outermost layer of your cell. So if you have cell A and you have cell B and you have cell wall and you have another cell wall, then the things that are between the two cell walls is going to be your middle lamella. Okay, and what it does is it acts as an adhesive which sticks those two cells together, and it gives the plant stability. So if you have two cells and they have their own cell walls. The thing that's actually sticking these two cells together in a compact way is going to be your middle lamella. Now, your plasmodesmata is going to be a channel through each of those, the cell wall and the thing. It's going to be a hole that goes through and links the two adjacent cells together. And that, of course, allows the transport of substances. And it's not just transport of substances, but it also allows communication between the cells. Communication between the cells is actually really important. So pits are going to be areas of just where the cell wall is very thin. They're arranged in pairs, so one on both sides, and it allows the transport of substances through cells. So if this was transport, if this was transport, and let's make this a bit thicker. If this was transport and communication, this one is going to be transport only. Okay, if I make this a bit smaller and remove that, now if I made this here and remove that, let's do that then. Alright, so yeah, now your chloroplast, we're gonna have 3 more. This is talking about like other, now we're actually leaving the cell surface membrane, cell wall, stuff like that. Now we're going into things which are inside floating around. So chloroplast, of course, is going to be the stuff that produces the food or the energy or whatever. So it produces the food. Okay. Flattened structure, a lot of similarities to your mitochondria here. Flattened structure, double membrane also. So flattened structure was in mitochondria. Well, I think it was. Double membrane was in mitochondria. Has inner membranes inside called thylakoid membranes. Now, your inner membranes over here was called your. Oh, actually, that's something that we can actually go into here for. So. Yeah, there you go. It's right there. So you have your mitochondria, oval shaped. Now this is great because oval shaped, it means that it's biconvex, which is also the same case in chloroplast. So this is also oval shaped, also double membrane. It doesn't matter what the name is. Inner membrane has inner projections called cristae, whereas in the other one it was called thylakoid membranes, right? And of course, since it has inner membrane, that means it has your inner, like, non-membranal areas, which is going to be called your, over here they're called your matrix, and over here they're going to be going on. They said membranes are stacked in some parts, so you have your membrane that's going around, and in some areas your membrane is going to be actually stacked one on top of each other to form areas of grana. Grana are linked together to form lamellae. So when you have multiple granas, you have another grana here, and another grana here, and another grana here, and you're going to be linking them together. That is actually called, the thing that's being linked, the thing that links them together is called your lamella. Multiple are going to be lamellae, and a single one of grana is going to be called granum. So now your lamellae is also going to be membrane. Grana's membrane, lamellae is also membrane, but grana is when you have multiple short membranes stocked up together, and you have the thing that joins up grana together is going to be your thylakoid membrane. Also, it's a thylakoid membrane, also known as lamellae. So they are separated by names depending on how they look like, what structure they have, what function they have. So this is of course the site where photosynthesis happens. Some parts of photosynthesis happen specifically in grana, other happens within the stroma. Um, the stroma is going to be just a fluid that's located inside here. Of course, when we looked at the mitochondria, we did know that there was going to be well, it's not mentioned here, but there is going to be an inner. It has its own cytoplasm, if I'm not wrong. So, um, you know, that cytoplasm containing the enzymes and whatnot, it's going to have it's going to be inside the chloroplast where it takes care of other reactions which photosynthesis will need. Now when you go into amyloplast and vacuole and tonoplast, right, so these are going to be amyloplast is, I'm pretty sure it's exactly what it says here. It's small, of course, and it's enclosed by the membrane. It has a membrane, and it basically just contains starch granules. Starch granules is just starch grains, just starch, basically. Just imagine a storage of starch. Storage of starch, and starch is obviously used to convert back to glucose when it needs for aerobic respiration, for energy. And vacuole and tonoplast. The vacuole, of course, as you remember, we've been studying it, but we kind of ignore it. All we know is that it contains, certain stuff like, it contains your cell sap, water, sugar, dissolved sugar, and stuff, whatnot. So it's a compartment surrounded by the membrane called the tonoplast. So the vacuole, of course, has its own membrane, as many other things do, and a lot of these things have their own names for their own membranes. In this case, the double membrane in the chloroplast, it doesn't mention what its name was, but the inside membranes are called the thylakoid membranes. Over here, the outside membrane of the vacuole was called the tonoplast. Over here, the outside membrane is not given. The mitochondria, looking all the way in animal cells, the membrane was called the envelope. Nuclear envelope was the membrane of the nucleus. So you have different types of names for the membranes of things. Now, specifically what this vacuole does, it contains cell sap, which is water, enzymes, minerals, waste products. This is important. It also contains waste products. This I think a lot of people forget this. They don't actually have. Well, I'm not sure why vesicles can't throw out waste products. I also remember lysosomes, right? They're vesicles containing your things. Of course, the lysosomes can't deal with anything inside, but they can deal with things outside. Because they contain like those hydrolytic enzymes which destroy stuff, but like whatever. The vacuoles keep the cell turgid as it grows, and it pushes the cell membrane with the cell. It pushes the cytoplasm with the cell membrane with the cell wall, so it keeps like a turgid pressure. This stops plants from wilting. It keeps them upright, keeps them stable, keeps them strong. Involved in the breakdown and isolation of unwanted chemicals in the cell. So, oh no, well, I am aware of the isolation part, not of the breakdown part. Uh, so those waste products, right, are the unwanted chemicals. They are broken down and they are isolated. Okay, yeah, well, no, see, they are broken down, but broken down means not removed or disintegrated into nothingness. There are still remains. There are still, what's left, and that's going to be isolated in the vacuole. So it's never leaving the cell. At least it's not stated here that it is. So it's stored somewhere in the vacuole where all that breakdown, all that waste stuff of unwanted chemicals stays. And the tonoplast is simply just what controls what enters and leaves the vacuole. Similar to as how the cell membrane here or the cell membrane here will also do the same exact thing. Okay. Now that we're done with the ultrastructure, I don't want to go into this just yet because the textbook does go into it in detail, and I do kind of like how they do it because they show more pictures and stuff. Um, oh god, I feel really awful. Okay. Okay, so let's just go through this a bit because I do kind of enjoy how they word out things here. So, um, plant cells are bounded by a cell wall. A cell as a jelly-filled balloon inside a small cardboard box. Okay. Cell wall, the cardboard box, important, blah blah blah. Now, see, here we go. It's made up of cellulose, and the plant cell wall is usually freely permeable. So, everything that dissolves in water can usually go straight through the cell wall. However, it does not act as a barrier to substances getting into the cell. Oh, sorry. Yeah, no, not however. It just doesn't act as a barrier. However, when suberin, alongside the cellulose, when suberin is added in the cork tissues and lignin is in wood, these compounds reduce the permeability and of the water of the cell wall, so that water and dissolved substances cannot go through it. So originally, you have your cell wall. Cell wall, I mean, I guess if it has the word wall in it, it kind of gives the idea that things can't really get through, but that's not true. Things can get through very easily as long as it can be dissolved within water. It can get through, and when it stops being able to get through is when your cell wall, which contains cellulose, is alongside that is added, either suberin or lignin. It depends on the plant. There are probably hundreds of more different things that can be added, but suberin and lignin are some examples of that. Now, the plant cell wall contains several layers. The middle lamella is the first layer and is made when the plant cell divides into two new cells, right? So now we're leading into what we kind of talked about earlier when it divides into two new cells. The middle lamella is the first layer that's formed. So when we have that cell and that first cell plate is formed and then it turns that into a cell wall, or cell membrane rather, the first thing in between those two things, of course, I mean, it has to make sense. You can't just start from one side and start making everything else and then you divide. I mean, sure you can, but that's not how it works. The first thing that's produced is the middle lamella, and the rest is starting to be formed, right? So, I mean, it makes sense. It starts in the center, goes out, right? That kind of has a kind of balance to it. So, the middle lamella itself is made out of pectin, which is a polysaccharide that acts as glue. Now, as we remember, middle lamella is used for, it's going to be used for it's going to be used for your adhesion, right? Adhesion, and combining sticking those two cells together and such. So that's what the pectin is really used, because it's the polysaccharide that acts as glue and it holds the cell walls of adjacent plant cells together. Alongside this, pectin has a lot of negatively charged COH groups, carboxyl groups, carboxylic acid group, by the way, and it combines with positive calcium ions to create calcium pectate. Now, this is also an important compound because calcium pectate will bind to the cellulose. Now, as you remember, it contains cellulose, the cell wall. So now, if this middle lamella is the first layer and it has cell walls right after that, and then after the cell wall, so let's say we do Control Z, we do blue for the cell. We'll do blue for the cell walls. Oops. So this is going to be your cell wall. This is going to be your cell wall, and this is going to be your cell membrane, and this is going to be your cell membrane. Okay. So what we're noticing here is that So what we're noticing here is that within the lamella, if you zoom in, this is the lamella, and right outside that is going to be, so this is going to be your lamella, this is going to be your cell wall. So you're going to have over here loads of negatively charged COOH groups, which then bind to the calcium with calcium ions present as well. And what that forms is calcium pectate, which then binds to the cellulose on the other side. So this is cellulose, 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 cellulose. and these are all molecules of calcium pectate. Okay, and if you want to know how calcium pectate is formed, as described earlier, it's just imagine COH group and pectin binding together. Sorry, COH groups and calcium ions. Pectin has the COH group, so pectin and the calcium ions, specifically within the pectin, is going to be your negatively charged COH groups. Now, right. So after it binds to the cellulose, microfibrils that we've mentioned a little bit already. Microfibrils, we mentioned them all the way in the Campbell textbook, but the microfibrils are coming from the kinetochores, right? So these cellulose microfibrils, wait, are they? Actually, since you said the word cellulose, it gives me an idea that it's not from the kinetochores, it's just a thing. I mean, either way, I think they're both microfibers which act in the same way for structure, and well, one is used to create structure, one is for structure. But anyways, the cellulose microfibers in the matrix build up on both sides of the lamella. This is kind of hard to understand, but what I'm kind of getting out of it is, if we looked at one side of the cell and we have the lamella, and then we have like the cell wall containing cellulose. So we have cellulose, right? And so the cellulose microfibrils which are present, these microfibrils and the matrix within it, all the stuff within the matrix and the matrix itself, will build up on both sides of the middle lamella. So of course, as we mentioned, along the microtubules from the kinetochore to the cell wall, we're going to be having, cell wall materials, cell wall components going down, right? And it's going to be reaching the middle lamella, where more and more, so it'll be reaching the middle lamella, where more and more stuff gets added onto the sides, forming finally, in a primary cell wall. So stuff will be added on and increases in width on the left and right side. And this is the lamella, of course. And so it's going to start forming a primary cell wall on the left and right side of the middle lamella. Now, secondary thickening might occur as plant ages, where your secondary cell wall is really, as it builds up, with the cellulose microfibrils laid densely at different angles to each other. So over here, your cellular microfibrils was formed in one direction only. Now what your secondary cell wall is it's going to be in loads of different directions, adding almost a second layer of structure, of turgidity, whatnot, right? It makes it much stronger. And of course, this is as it ages, as it'll go from these smooth, straight lines into something that's more random. So, as it's in different angles, this will make the composite material of cellulose microfibrils, much more rigid. Cellulose, hemicelluloses help. So this is alongside the different angles. We have a different compound called hemicelluloses, which harden it further. Okay, and in some plants, particularly woody perennials, perennials, lignin is added to the cell walls to produce wood, which makes it even more rigid. Of course, as it becomes less permeable, it becomes more rigid. So there you go. As we know, lignin is added to things because we saw lignin once before. If you found it recognizable, that's where we saw it. And you know, there's many long cells. So it makes them more rigid. And within the cell structure of a plant, there are many long cells with cellulose walls which have been heavily lignified. That is a beautiful word right there, lignified. So yeah, lots of long cells are present with cell walls which have been lignified to just make them more rigid. These are going to be called plant fibers as we go on, and people use them for clothing, building material, ropes, and paper. So this is kind of connecting section 4.8, 4.5 with what we're talking about here, because when we have cellulose cell walls, which is lignified, turned more rigid, turned more impenetrable, turned more, the impermeable, right? It's turned into these more and more in these different things. Then it starts turning into plant fibers, which can be used for actual manufacturing. Now, the cellulose chemistry is honestly really simple. I studied a long time ago. I don't know, maybe the way I studied it was really, I just find it super easy. This is going to be your normal glucose right here. This is going to be alpha glucose. I mean, there's no normal glucose, but this is going to be your normal glucose that you're going to be studying a lot in Unit 1. And this is going to be your alpha glucose, which is going to be the same thing, except only the right side is flipped upside down, right? Now, once that's happened, the right side is flipped upside down, that means that, well, first of all, you need to understand that in cellulose, there are only beta glucoses available. And you'll see why right now. Actually, wait. But there is no reason why. Hold on. So beta glucoses lined up, right? And if you remember, for glycosidic bonds, what do you need? For glycosidic bonds, you need you need two OH groups. You need two OH groups because you'll find an H and an HO, and you'll get an H2O from there, and you have the glycosidic bonds of an O. You can't have one H and one OH. That's not going to work because they'll just form water and there will be no O for an actual bond to be formed. So you need two OH groups. And as we see here, if we had another of these, we would be getting an HHO as we move on to the other stuff. And of course, we can't form any glycosidic bonds here, so we have to invert this into an OHH, as in we have to flip this around, forming an OHH, which can form a glycosidic bond through it. Now, so now the glycosidic bonds can be made after it's been inverted, and the linking of these can mean that hydroxyl OH groups stick out on both sides of the molecule. Oh, what does that mean? What that means is especially in small molecules, you can have OH groups which are bonding with these, forming hydrogen bonds with other ones. But it's not really the ends of these, it's usually these within, which isn't really a special scenario that happens with glucose as well. Cellulose do not coil or spiral, you can imagine, because since they are inverted, they go like this, they go like this, they go like this. They just form a straight thing without any spirals. They remain as long straight chains. And whereas, of course, glucose will form spirals, and the difference in structure between starch and cellulose gives them many different properties. Just a little fact is, since it's joined, okay, now we did a glycosidic bond. What is the name of the glycosidic bond? Of course, it's going to be 1, 2, 3, 4, 5. Is that 6? I'm not sure. Of course, it's going to be the fourth carbon over here bonding with the first carbon over here. So we're having a 1,4 glycosidic bond. So 1,4 glycosidic bonds, 1,6 glycosidic bonds. Why are we saying 1,6? Let's look at this. Sorry, it's inverted, right? So this would be 1, 2, 3, 4, right? Because it's inverted, so we're going like this, and this one is going like it's going like this. So it's going clockwise. Since we inverted it, it's going anti-clockwise. Now, so what is 1,6 bonds? 1,6 bonds is going to be formed as a result of your branching. One, and what is a 6? 1, 2, 3, 4, 5, 6. So you're going to be having things which are joining to another thing, like in that kind of sense. However, the worst one, the 1,4 glycosidic bond, the special one, the one that happens the most, cannot be broken by enzymes. Well, they are all broken by enzymes, but that enzyme is not present in humans, so we can't really eat them. Only ruminant animals can eat them. They have cellulose digesting enzymes for bacteria living inside their gut to digest it. So that's going to be what your cell wall is truly made out of. Now, these cellulose molecules, as you can see here, these are beta glucoses, but they join together to form cellulose. You'll have cellulose molecules which will form these huge fibrils, which then you can actually be able to see underneath an electron microscope. Cellulose fibrils are deposited in layers. Of course, the primary means that it's in one direction, secondary means it's in random directions and different angles and stuff. These cellulose fibrils are deposited in layers which are held together by a matrix of hemicelluloses, of course, which will make it much stronger or hardened. They act in the same way as glue, blah blah blah. Mannose, xylose, arabinose are used in this process. They're going to be some of your hemicelluloses and short chain carbohydrates used to glue them up together. The glue was, what was the glue again? The glue was the thing itself, the middle lamella, right? But this is just more. So the combination of the cellulose microfibrils in the flexible matrix makes a composite material combining the properties of both of these materials in the plant cell wall. The cells are turgid most of the time, giving strength to support the plant in a vertical position, yet the plant can wilt when fuck. Okay, what else? I don't really know what the point of this is. So of course your plasmodesmata is going to be like that bridge between it, used for transport. There are some terms which are kind of cool here. The plasmodesmata itself is just kind of the bridge so that they can communicate, so that they can transfer materials. But there's going to be an interconnected cytoplasm within these two cells called the symplast. Okay. Now the cell walls are thinner in the regions of plasmodesmata. Of course, it has to be. Sorry, we skipped the important part here. So plasmodesmata is a bridge, but the bridge is not separated. Like the two cells do not separate completely here. Like it's not like this. Okay, it's not like there's literally a free hole. There is stuff between here, and that stuff is going to be threads of cytoplasm remaining in between them. There's going to be threads of cytoplasm, of the cytoplasm itself. Yes, it sounds weird, threads of cytoplasm, but it is cytoplasm threads which remain there, and the threads pass through the gaps of the newly formed cell walls and signaling substances. They pass through the gaps of the newly formed cell walls, and signaling substances, like the thing that's going to be used to communicate, can pass from one cell to another through the cytoplasm. The interconnected cytoplasm is actually called the symplast. So when you have the hole, and it's not truly a hole because you're going to have certain threads of cytoplasm within there called the symplast, which allows stuff to weave through the holes and actually get through and communicate or do whatever it wants. When you have your second thickening, hemicelluloses lignin are deposited, making it thicker, and the process doesn't happen, leaving thin areas. So wherever you had, plasmodesmata, after the secondary cell wall is made, after the hemicelluloses make it hardened, then all you have is just a kind of dip, which is called a pit. It still says that things can move there. It says here there is no cytoplasm in the xylem cells, but pits within them allow water to move between the xylem vessels. So you're going to be having two xylem cells, for example, one xylem and one xylem, and you're gonna have pits within them, which allows water to actually move through the plasma, move through the pits. With his iphones, man, goddamn, I'm tired. After we finished talking about cell walls and whatever, there's the plant organelles that we have to go into extra, which I don't think we really need to do, other than just being able to know, being able to recognize what they look like. I don't think it's very necessary to go through this. So that takes us to plant stems, which we just use this. So plant stems are made up of different things from just my prior knowledge, right? We're gonna have your xylem. You're gonna have, I think, your phloem like this, and then you're gonna have your sclerenchyma. You're gonna have your parenchyma, then it's sclerenchyma, and then it's parenchyma. Bro, I forgot what it's called. It's called collenchyma, right? So you're gonna have those things. I'll put this closer. So yeah, you're gonna have those three things. For some reason, that's not only sclerenchyma is here, not parenchyma. I'm not sure what's going on. So, anyways, talking specifically about xylem vessels as it's used in this plant stem specifically. So we're going to be talking about GCSE stuff, like the structure and function of those adaptations or whatnot. And so it's mostly going to be GCSE stuff for xylem and phloem, but for sclerin, of course, that's going to be new. Function of xylem: transport water, obviously, up the plant, specifically, not around the plant. It's only upwards, and it's also used to provide support. That's why it's going to be made out of thickened stuff, especially lignin, which is obviously going to be impenetrable or impermeable. So it makes sense, Cell walls are made out of lignin because you don't want it to be exiting only through the pits. You want them to exit. Okay. They're very long tube-like structures from dead cells joined end to end. Tubes are found in bundles. Bundles as in you're gonna have another, you're gonna have another, you're gonna have another, you're gonna have another, all grouped together. Lots of tubes together going down. The longer they are wide, so these are longer and thinner than a normal average cell. So maybe perhaps that would be a normal cell. So you can obviously see that it's much longer, and also it is also thinner in comparison, right? And there's reasons for that. Of course, you want it to be carried a longer distance, and you don't want a greater area because you want high pressure so that it can actually go up. So those could be reasons. They also have no cytoplasm, of course, no end walls, so that it's an uninterrupted tube, making water and minerals pass up more easily. So that's the main function of all of these. It's just so that water and mineral ions can pass up more easily. Walls are thickened with lignin to support it. Easy peasy. Water and mineral move into and out of the vessels to the pits in the walls where there is no lignin. Of course, as you remember, the pit is just that kind of area with less cell wall concentration. Cell wall decreases, and so water can actually pass through the xylem in those areas. Now let's just go to phloem for a sec. Okay. And actually, no, this is too boring. So let's take sclerenchyma. So the function of sclerenchyma straight away, we're just talking about the function rather than the support, rather than the properties. Is to survive support, like mechanical support. Okay, sclerenchyma. They're not really involved in transport. So similar to xylem phloems, they're made out of dead cells, which which are run vertically up. Okay. And these cells are longer, it's wide, they have a hollow numen, but unlike xylem, they have end walls. Because again, they're not transporting anything, so they're probably going to have end walls. Now the cell walls are also thickened with lignin, of course, but they do not contain pits, again, because they're not used for transport. And they have more cellulose, of course, because they're used for structure, to be more rigid. You could probably think of it also having more hemicellulose. Probably will have that adaptation. You can imagine. Function of phloem tissue is obviously to transport organic solutes, mainly like sugars, like sucrose, which is going to be coming from your leaves, right? From where they're made in the plant. So that could be your leaves, that could be your flower itself, who knows, maybe even your stem, because they also contain chlorophyll. Some stems are green. To where they're needed. This is known as translocation. So like xylem, phloem is also made from cells arranged in tubes, of course, because you're transporting them. The best to always do that in like pipes or tubes. But unlike xylem, it's purely a transport tissue and is not used for support. So xylem is also used for support, so that's why it's having dead cells, that's why it's having lignin, that's why it's having, um, you know, those two, I guess. This one, absolutely not, no transport whatsoever. You can imagine that's because, um, you can imagine that's because food is more vital, and you need way more like areas of transport outwards in the phloem, which like kind of prevents it from being a support because there's just it's there's just too many holes for it to become a supporting tissue, so, or like a strengthening tissue. So it's more like just a transport tissue. Now, phloem tissues contain different types of cells, including sieve cells, sieve tube elements, and companion cells. This is GCSE stuff. Your sieve tube element, as you can see, kind of like over here and here. Or rather, no, this is a sieve tube element right there. Or am I wrong as well? I'm not sure. Anyways, your sieve tube elements are living cells and are joined end to end to form sieve tubes. So these are living cells over here, and they join end to end to form a sieve tube. And these sieve parts are just the end walls, which have holes or sieves, allowing the solids to pass through. And unusually for living cells, sieve tube elements have no nucleus, a very thin layer of cytoplasm, and few organelles. So that actually, this bluish area is actually going to be your sieve element. It's a living creature, and it has nothing except for cytoplasm. And so yeah. And what the sieve, what this element really does is it joins together with other things to form this kind of plate full of holes, which allows there to be a sieve which the food passes through by. And as since sieve tube elements have no nucleus and organelles and stuff, it has very few, but mostly none. The lack of nucleus means that it needs an element. It needs something to survive on, because it cannot survive on its own. So that's where the companion cell comes in to accompany each sieve element. And what the companion cells do is they carry out the living functions for both themselves and the sieve cell. For example, they would provide energy and transport of solids. Now talking about vascular bundles, this is like the real stuff, right? So you're gonna have your xylem, phloem, and sclerenchyma together in one bundle, but you only need to know the positions in the stem. This bundle, since they're tubes and they go throughout, you can call them fibers, right? So all three these vessels, tissues, and fibers, they're found throughout the plant, but it doesn't matter where they are. They only their position in the stem is always the same. So in the stem, xylems group together with phloem tissues to form vascular bundles. So as you can see, if I can zoom a bit, we have, or we could even go into here, or even a better image is actually here. Yeah, so this is a wonderful image, and we can see your sclerenchyma fibers. Your sclerenchyma fibers, you have your phloem, you have your cambium, which is just the thing between the xylem and the phloem, and you have your xylem, and then you have your so this is going to be a vascular bundle, and there's going to be multiple. There's going to be one here, there's going to be one here, and so as you see them repeating on and on, around the stem of this, young plant. Notice how the xylem and the phloem is surrounded by the sclerenchyma and surrounded by the xylem, and the xylem is the biggest one, with the phloem being the smallest one. So these are just some structural things to take note of. Over here in this image, we're seeing sclerenchyma fibers on the outside. We're seeing phloem tissues being surrounded by the xylem tissues and the sclerenchyma. And the thing they even drew the middle line, that's going to be your cambium. If my understanding of cambium was correct, it's just kind of like stem. It's like unspecialized cells, which kind of act like stem cells, which specialized between these two, to produce more and more xylem and phloem tissues or cells to form tissues, so that they grow as the actual width of the stem actually increases. Because as the stem increases, you need these vascular bundles to also grow in size, right? So that's what the cambium is actually used for. Now, if you look at longitudinal cross section of the stem, this is a transverse, and the transverse is obviously at a right angle to the length. As in, if this is the thing, we're taking it to a right angle to its length. So this is length going this way, and we cut it this way, that would form a right angle. Now longitudinal cross section are taken along the length, so if it's going this way, we cut it this way as well. So now we're looking at it kind of like halfway. Okay, so now what this would look like is we're seeing, what are we seeing here? Okay, so we've cut it twice actually. Well, no. See, we're seeing one vascular bundle here and one vascular bundle here, and so we're seeing the xylem on the inside, the phloem in the middle, and the sclerenchyma in between, on the outside. So sclerenchyma, phloem, xylem. Okay. Now dissecting plants. If you look at a plant under a light microscope and identify the xylem vessels, seed vessels, and sclerenchyma fibers, we may need to do that, or we can do that, and you can look through pre-perforated slides to look at, or you could dissect the stem and prepare a section of the tissue yourself. If you're dissecting the plant stem yourself, you can use this method. So again, we're trying to get a sample of a stem so that we can look at three things: the xylem, the xylem, the phloem, and the sclerenchyma tissue fibers. So you're going to take a sample, a scalpel, and of course cross a cross section of the stem. Cut a cross section. Now that cross section could be longitudinal or it could be transverse, and you want to cut them as thinly as possible because you're using a light microscope and you have light passing through. So you need it to be as thin as possible so that it can actually go through. Now, so using tweezers to gently place the cut sections in the water, and then so you want them to stay moist because I guess that allows the water to get absorbed. The light to get absorbed nicely or like faster, because you do need to put it into a solution. But this is an interesting question. Why do we need them to be put in water before we put them into a light microscope? Makes it easier to see is my idea, or it reflects the light so you can actually see it better. So now transfer each section, including a stain. This is the stain that we used in my class, and leave it with that stain for 1 minute to soak. This contains lignin blue-green, which allows you to see the position of the xylem vessels in the sclerenchyma fibers. Of course, it tells you by its name that it contains lignin, so of course the supporting things will obviously contain lignin. The phloem cells will not contain lignin. They would appear purple, while everything else turns blue-green. Now rinse off the sections in water and mount each on top of a slide. So after we've let it soak, and then we put some stuff on it, and then we rinse it off with the water, and then we put it into this slide. Prepare the slide under a microscope and adjust until you can see a clear image. Make a labeled drawing. So let's see, what is X and what is Y? So if you look into the image closer, X is on the outside, so that's going to be your sclerenchyma, and this is going to be on your inside, so that's going to be your phloem. Now, starch, cellulose, and plant fibers. So we've talked about this partially, but this is starch. Now, starch, we discussed it heavily in Unit 1, and all cells, or at least all cells that I know of, get energy from glucose, right? So they need to break it down using aerobic respiration to release energy in the form of ATP. Now, starch is the source of glucose, at least in plants, right? And to break down starch, well, you need to perform hydrolysis of it, because you want to break down, because starch is a mixture of polysaccharides of glucose. You have both alpha glucose, amylose, and amylopectin. Amylopectin and amylose are almost the same thing. It's just amylopectin is just branches of amylose together. So amylose is basically just all this. If you study glycogen in paper 1, you will notice that this is quite almost the same exact explanation for amylopectin and amylose. You have your long unbranched chain of alpha glucose. The angles make a coil structure, almost like a cylinder. This makes it more compact. Good for storage. You can fit more into a small space. If you had a long chain and you had a small box, you wouldn't be able to fit that in. But if you had the long chain curled up into a bunch of things, guess what? You can fit that in, and not this. Okay? Amylopectin is a long chained branch of alpha glucose, and it has side branches which allow it to break down more quicker, break down more easier, and so energy is released more quickly. That's the main point. Starch is insoluble in water, so it does not affect the water potential. So that means it doesn't cause water to enter cells by osmosis. The vacuole, we didn't state this, the vacuole also does not affect water potential. So when stuff goes in, that is actually not going to affect anything about the cell, so the cell will continue taking in stuff, and after that stuff is going to be processed, and waste products are going to be put in, or water is going to be put in, or sugar is going to be put in, that's produced as a result of this whole aerobic respiration thing. It's going to be put in the vacuole, which will basically just make it forgotten in terms of water potential. And so more and more and more stuff will go in. This will get more and more and more and more filled, allowing a greater pressure on the cell walls, because it grows in size, and it causes a turgor pressure as it pushes the cytoplasm towards the cell wall. Now, starch is insoluble in water, so Now we're talking about cellulose. It's going to be basically your cell wall stuff. Your beta glucose 1,4, it reverses every single one, and there's going to be hydrogen bonds between each 1,6. Hydrogen bonds are straight, so cellulose chains are straight. Between 50 to 80 cellulose chains are linked together by a number of hydrogen bonds to form strong threads called microfibrils. So when you have 50 to 80 chains, when you have like 50 to 80 chains, imagine like a 3D structure here, right? You have 50 to 80 of those chains. This is 1, this is 2, this is 3. Now imagine 50 to 80 in a little tube. That is, and of course they're joined together by loads of hydrogen bonds. Now that entire tube is, it's a thread basically, right? That's what you would count it as. And that thread has a more scientific name called a macrofibril. That microfibril will turn into a fiber, I believe. Maybe there's something in between, I'm not quite sure. So yeah, that provides structural support for the cells as they increase the cell strength. So this is just to explain why is cellulose used as a structural protein, structural, no, it's not protein, sorry, structural carbohydrate. Why is it used as structural? Because it has hydrogen bonds. It can form these microfibers, and those microfibrils can be able to form fibers which will strengthen the plant. Now, more specifically on fibers, these fibers will be again made out of long tubes of plant cells. That could be your sclerenchyma, that could be your xylem, made up of dead cells, made up of lignin, made up of cellulose, right? They're strong, makes them used for carrying loads, which could be ropes, fabrics, So we're talking about useful to humans because of one reason, and that's they're strong. So we can see that they're strong, so that we can use them for heavy carrying objects like ropes and fabrics, right? Like hemp. The hell is hemp? Anyways, they're strong for a number of reasons. We only need to know 2. One is because they contain the cellulose fibrils that we mentioned in a net-like arrangement. Net-like meaning that we have them going in different angles, which is actually what we referred to when we were talking about the secondary secondary membrane, secondary cell wall, right? Because the secondary cell wall means you have another layer of macrofibrils which are going in opposite random angles, which causes layers of cellulose microfibrils to kind of overlap on each other, causing greater rigidity, greater strength, and overall that is allowing it to have greater use outside of just plants and stems. Now the strength of the microfibrils and their arrangement gives the plant fiber strength. This is obvious. And the secondary thickness, oh well, that's okay. That's mentioned. That's the thing that we mentioned. This is very vague. Perhaps the textbook will have it better. Oh yeah, so here just mentioning it. Cambium is your layer of unspecialized cell which divide, giving more unspecialized cells which both the xylem and phloem use. So it's a layer of unspecialized cells and gives rise to the xylem and phloem. This is the xylem, this is the imagine this is going to be your sclerenchyma, so this is going to be your phloem. So it'll give rise to phloem, it'll give rise to xylem as it grows in size. One second. Okay, there you go. So sclerenchyma is going to be a type of modified parenchyma found in tissues. Empty tube, simple pit, We were not talking about any of the structure regarding your sclerenchyma. In the spec, I think we did mention that we need to know the ultrastructure of sclerenchyma, no? Understand the structure and function, understand how the arrangement contributes to physical properties of xylem vessel, contributes to the physical properties of xylem vessels and sclerenchyma and plant fibers. That can be exploited. Yes, that makes sense. However, again, similar differences between structures of the position in the stem and the function of sclerenchyma fibers. Okay, so we need to know the differences between the structures of sclerenchyma fibers, xylem vessels, and phloem, which we did, but just not in as much detail. Because when we talked about sclerenchyma, did we have a diagram? I don't think we did. No, we didn't. Well, this is what it looks like. You have your lignified secondary wall, as you would also have it for your your xylem, or your normal cell wall in case you wanted to make it more rigid. It has empty tubes and no living contents. However, it does have end walls, right? So that's going to be a sclerenchyma cell. This is going to be a sclerenchyma fiber, and that's going to be the thickened sclerenchyma tissues, as you can see them over here, practically, surrounding the entire thing. Of course, this entire thing is going to be your vascular bundle. My point being, however, is what is going on with the fiber? Why do they have bits? I thought they were not supposed to have bits. Let's see. So they mentioned here that sclerenchyma. I'm not sure actually. So, found around the vascular bundles in older stems and leaves, sclerenchyma have strong secondary walls made of cellulose microfibrils, positioned at right angles to each other. So again, we have, strong secondary cell walls made up of cellulose microfibrils at right angles. So again, think of your primary as in one way and your secondary as in your second way. Okay? And some sclerenchyma fibers make fibers. Some sclerenchyma make fibers. Some very long cells, often found in bundles or cylinders outside a stem or root. So what they're trying to say is they can form different types of things. They could form a fiber. They could form just a cell. Lignin is deposited on the cell walls of these fibers. When the fiber is lignified, cell contents die because water cannot pass through it, so they become hollow tubes. Okay, they don't talk about why the pits exist, which is confusing. Okay, here we go. You see how it's pink? That means that they were using the blue lignin. The thing that turns lignin blue-green. So you see that there clearly. How the things which contain lignin in your xylem and your sclerenchyma is going to be green, and everything turns pink. Else turns pink. Even your cambium is slightly green, but I guess it's just because it's unspecialized, so it could have anything. anything here that's kind of interesting to look through? See, this is going into another route. So here we have another route. So we're seeing airspaces. We're seeing the center. Okay, so in this case, we have in this kind of route, we are having only one xylem. We're not having multiple on the sides. We're having only one, and whoops. So what that leads to is again just phloem in the center. Phloem is not in the center. Xylem is in the center. However, there's pericycle, endodermis, and cortex made of parenchyma cells. So this is just a different kind of thing. So here we have xylem going kind of like this. Phloems on the little sides over here. So okay, my guess is if you had it like this and you had this being your xylem, this being your phloem, that means your phloem is entirely protected by only xylem. Now I would think that whatever's over here and whatever's over here and whatever's over here is much stronger than this xylem. So I would imagine that again they would protect it in this kind of way where you have two of them pushing against each other to form a better kind of protection. That's what I would kind of try to visualize it as. Yeah, so here we have a very nice diagram. Now we have not looked through the sclerenchyma yet, or nor the parenchyma, in fact. And I also did want to slowly just read through this. So, flowering plants that we're studying, which are called angiosperms. Imagine sperm as your, reproduction form, angio just being whatever, and so the flowering plants do reproduce in that manner. So they do need specialized cells for that. And so they can be monocotyledons or monocots or dicotyledons, which is dicots. So monocots are dicots, okay? And what those are, what characteristics they have is that monocots, such as grass, have long, narrow leaves, whereas dicots have leaves with stalks and blades, which is petioles. So again, monocots have more like long, narrow leaves, so like your grass, right? But your dicots will have blades and stalks. So you're gonna have blades, I think that's what a blade is, and stalks, which I'm pretty sure is like very thick. Your very, very fat leaves. So you're gonna have both of those in your dicots. The mechanism transferred through both are the same, but there are differences in the distribution of the xylem and phloem in their roots, stems, and leaves. Only dicots plants are described in this book. So, yeah. That's what a dicot is basically. So the only difference between them that I should really know is the fact that they will have some difference in how the distribution of the distribution of the xylem, phloem, and sclerenchyma are going to be. Now, quickly, I just want to go through what the parenchyma and blah blah blahs are. So over here, collenchyma, it says that it's also used for strengthening. And sclerenchyma is in fact not even here in this image. So compact parenchyma is what I'm supposing to be. You're including. Okay, so this is what I want to be looking at. So parenchyma is made up of thin-walled cells used as packing tissue. So you're having your cells used as packing tissue. Is this 7 by 10? Yes. So using this is parenchyma transverse section. Okay, and these cells are very metabolically active and may be used for very many functions. They may be used for storage. When turgid, they help to support the plant. Air spaces allow gas exchange. Water and minerals are used for are transported through these walls and through living contents. Parenchyma also forms the cortex in the roots and stems. Okay, real quick, I just want to know what a cortex is better. What is a cortex in bio terms? So the cortex is the outer layer or region of an organ or structure. When we have cortex in plants, it's the layer of cells just located below the epidermis, outer skin in stems, and roots. So it's just in between your epidermis and your actual root itself, like the inner parts. And it's involved in the transport and storage of nutrients and water. So if your epidermis was somewhere over here in terms of this going outwards, then your matrix would be probably somewhere like in this region, right? And so that's why you have parenchyma all over here, which store your water and whatnot, for later uses. Now, it forms the cortex and roots and stems, and of course, since it's an outer region of cells, the pith, I don't know what that is, made up of similar cells, but the name is given to a central region. Parenchyma also contains chloroplasts, where it is modified to form the palisade and spongy mesophil. So the parenchyma can also be modified to form the palisade and spongy mesophil cells when needed. So each of these are parenchyma cells. Now we have collenchyma and we have endodermis. We are not talking about sclerenchyma. This is so odd to me. Sclerenchyma is mentioned here. Sclerenchyma is also mentioned here. So over here we're talking about collenchyma being a modified form of parenchyma with extra cellulose. Deposited in the corner of cells, this provides extra strength. The midrib of the leaves contain collenchyma. So I suppose since the stem contains parenchyma, the midrib of the leaf will contain collenchyma. Is that? I'm guessing that's just how it works. Is there a section regarding stems? Because this is the midrib, of course. Structure. Huh. I suppose not. I'm not very sure why. I'm confused. Now they're talking about the xanthan tissue. Oh yeah, dude, they're really talking about it. Hold on. Okay, now, okay, so. Okay, bro. Alright, so that's where it really gets into the stem. Alright, sure, fine, cool. So what are they talking about up here? No idea. So collenchyma is just modified parenchyma meant with extracellulose for structure or for mechanical purpose. So as we go down, we're seeing the actual vascular tissue, right? And it contains more than one type of cell, and together they form the vascular tissue. Vascular means that they have tubes for transporting fluids. They have xylem vessel elements, lignin, mechanical support, yep. Xylem foam, vascular bundles. Outsides of these bundles are made up of sclerenchyma fibers, which have extra support. Sclerenchyma fibers, like xylem, are also long dead empty cells with lignified walls, but however, they have only a mechanical function and do not have to support any other function like transporting water. So in this picture you have cross section of a leaf, I think. Okay, now so you have sclerenchyma cells itself. We're having a picture of those. So we are seeing sclerenchyma. You're seeing the secondary wall of the fiber, and we have lumen, and we have the phloem. So we have the phloem over here, sclerenchyma over here. So you can imagine the xylem being somewhere over here. Okay, and I'm trying to see what they're trying to tell me. So the lumen is the whole. The thick wall is going to be your secondary, and also obviously your primary, or your wait, no, it's called just your normal cell wall, right? I forgot what it was called. Your normal cell wall, and then your secondary cell wall. So that's also going to contain your microtubules of cellulose, all that jazz, and it's going to be all packed into this one thing. So that's going to be your sclerenchyma, all of it. Now this is going to be a better image, and see, they do have pits that connect to each other. I don't know why they don't talk about this. So there's no intercellular air spaces present. However, in the cortex there was, because there are other uses of your parenchyma cells when they're in the cortex. So imagine this is a thing, okay? You have your vascular bundles, okay? You have your sclerenchyma, okay? Since we're in the stem, there's going to be no collenchyma. However, or perhaps there will be, but it's a mixture. The point being is that collenchyma and sclerenchyma are used as structure in their own specific areas. But there's still a bunch of space here in terms of cortex. This could be, you can imagine there's also another plant stem here with a thing in the middle, which is going to contain your xylem going in like this, and your phloems here, and so you're going to have your sclerenchyma, and then you're going to have all this remaining space. It's cortex. So that cortex is going to contain your palisade cells. It's going to possibly contain a mixture of collenchyma cells. And there's going to be the biggest difference is the sclerenchyma here has no gaps. It's all, there's no air spaces, there's no gaps. It's just these empty lumens with no water, no nothing. And now a cross section of this would look like this. So you can clearly see your lumen being this wall over here, or this hole over here that it goes up by, and your lignified is going to be this lignin wall, secondary wall. So imagine this is your primary, this is going to be your secondary. It's like thicker. That's it. And your overlapping tapered end walls. So of course there's going to be end walls which taper, as in if it's going to go like this, there's two tubes. There's going to be one that kind of goes in like that. Okay, so it tapers into the next cell wall, producing an end to this one area, an end to this one area, a start to this new area. So a better, easier viewing thing is this is your lumen. This is going to be your simple bits. You're going to see bits that are going to be like probably like this. I don't know. That allows stuff to go through. I don't understand the function of this, but I really don't know. Overlapping tapered walls, so it goes in like this, and it tapers and into two, forms a new hole, but ends one hole. It's a whole maze of stuff. Transport of water itself, I'm not very interested in right now. This is going to be a leaf structure. If we just want to look at it, this is looking like a monocot because it's very thin, but it's a blade, I believe. So we have, you have your edge, your upper epidermis, you have vascular bundles. Oh wait, sorry. This is a monocot. This is a transverse cross section. We've cut through the leaf going like this, and we've cut through it in this way. And so we're only seeing a brief line of it. So we're seeing the upper and the bottom. And in the middle, this is about, I would say, the midrib. So you would have a lot of collenchyma here, a lot of collenchyma around here. And you're having these lines. Not sure what these lines are actually. I would say they're things that connect the membranes, but I might be wrong over there. Your specialized membrane cells, which are going to be your guard cells, your stromas, cuticles. Cuticles is going to be your waxy cuticle on the top to hold down on the rain. And yeah, I mean, dude, we learned this diagram in like grade 1. I mean, this is genuinely such an old diagram. This is regarding more closely the transpiration stream, I believe, which I'm not going to be looking into just yet. Are we not going to be talking about any more of the xylem and the phloems? There we go, come on. So yeah, here we have the symplastic pathway, apoplastic pathway. This is kind of what I wanted to see. So yeah. Oh my god, no. No way I just did that. Annoying. Yeah, give me like 5 minutes. Dude, that is genuinely an annoying thing. So. I don't know why it's like this. It's like, it seems very odd, but can I not click on this? There you go. Okay. So we're seeing so from the xylem to the leaf, right, or from the xylem across the leaf, water evaporates from the cell walls of mesophyll cells, blah blah blah. So okay, fine. So okay, maybe I do need to know this then. Because this is okay. So now this is in roots, and this is in leaves, and so what? So first we looked at stems. Now we're looking at leaves. Next we're looking at roots. So in our bio textbook, or in the specification at least, do they mention that? They mention... Not really. That's not really there. And they only mention the sclerenchyma, which probably means that we're only looking at the roots right now. But there is an image of the midrib, dude. How confusing is that, no? Looking at here, it's just gonna be another reference. After this, we're talking about cellulose microfibrils, talking about uses. Just uses and 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 uses. And the biotic stuff. Okay, well that's pretty much everything I need to know regarding this. The main concept of parenchyma of the cells and whatnot. So I think I'm done. I'm done. Oh no way, the music ends there as well. Oh no, it looped a long time ago. Okay. Yeah.